Nervous tissues are very specialized to activate many processes by sending electrical impulses that cause chemicals to be secreted at synapses and is done very fast and can be done over very long distances. Now it's pretty basic and most of you already have a general understanding of that. The problem with nervous tissues in introductory histology lab is they can appear very diverse under a microscope and there are many staining techniques to complicate things. So we're going to try to keep it simple and just identify the tissue type as well as identify a few different structures. So here are a few examples of how diverse it can appear. Here we have a cross section of the spinal cord where we can see gray matter containing mainly cell bodies and the white matter containing mostly axons. Versus a chunk of the cerebellum here, which is part of the brain, we can see some complex folding involved there. Then we have a nerve and cross section over here where we see mostly axons transmitting electrical signals and the surrounding insulation of myelin. So let's keep it simple and go over the basics on how to identify nervous tissue. Here's a basic diagram of a neuron, and here are three different structures we're going to be locating and identifying to help aid us in determining if a sample is nervous tissue. Now, first of all, we want to be able to locate the neurons. The most obvious structure in the histology slide is arguably the cell body of the neuron, also known as the soma. So that is roughly this region right in here, the soma. Now, you might also hear this referred to as the perikaryon, peri meaning around, karyon referring to the nucleus, and the nucleus is that portion right in here. So there are some other important structures that are unique to these neurons, and those include the axon as well as the dendrites. So our axon here is this region, and our dendrites are these structures right here. So as far as the axon is concerned, it sends electrical signals away from the cell body, whereas our dendrites are going to be receiving signals from other neurons and sending it towards the cell body here. Next are our glial cells, and they're also known as our neural glial cells. They can be thought of as our support cells, and they have various different names based on their function and location, but they are all there largely to support the neurons. Some functions include recycling neurotransmitters, producing myelin for faster nerve conduction, and some are involved with immune response, so they're pretty important. In the peripheral nervous system, sometimes abbreviated the PNS, those glial cells are known as the Schwann cells and satellite cells. We won't be looking at those in this video. Instead, we're going to focus on the CNS, or the central nervous system. Now this image over here contains our four different types of glial cells: microglial, astrocyte, ependymal, and oligodendrocyte. Now, based on the staining method, we're mostly just going to be able to see the nuclei, and we're going to consider them glial cell nuclei, and we're not going to differentiate between the four. Other than the ependymal cells, due to their location, they're rather difficult to distinguish with light microscopy, so we're just going to call them glial cell nuclei. So here we are zoomed into 400 power looking at a cross-sectional view of the spinal cord. This is a silver stain and it does an excellent job at outlining the soma as well as the nerve processes, both axons and dendrites, dark brown. So here we have an example of a soma, and contained within that soma we have a large nucleus. So soma and then nucleus of a neuron. Then we can see these neuronal processes stained in this dark brown color. They're either axons or dendrites. However, we really can't determine the difference based on the staining method. And most of our samples we won't be able to either. So just neuronal cell processes, and it gives us this overall shape. You can see there's one right here, there's another one over here, another one. They're quite all over the place. Next, we're going to be looking at our glial cells. And our glial cells, it's going to be hard to see because of the color that they are highlighted in my diagram down below, or my table rather. But we have glial cell nuclei. Here's one there, another one, another one. You can look all over the place, and it's pretty obvious we have a lot of these circular structures. Those are our glial nuclei. Now, note that they clearly outnumber our neurons. And conventionally, scientists thought that these glial cells outnumbered neurons 10 to 1. And obviously, we see that here. It was conventional wisdom among scientists long ago, but newer studies are showing us that it's closer to a 1-to-1 ratio. But that's beyond the scope of this video and how they determine that. But it's fairly obvious with our slides that glial cells are going to outnumber neurons. So how do we identify this as nervous tissue? First, we look and we find a fairly large soma containing a nucleus and these beginnings of these neuronal processes here. And then we identify that there are nuclei, these smaller ones, that greatly outnumber our neurons. Note also the size difference between a neuron and our glial cell, at least the nuclei, we can clearly see that as well. So that's our first sample. Let's look at two more. So again, we're at 400 power and we're looking at a cross-sectional view of the spinal cord. However, in this case, this is a traditional H&E stain, and as such, the nuclei are going to stain dark purple. Although not as obvious here, that spot right there in the middle is staining darker. So to start off, we're going to outline the soma, or the cell body of a neuron, and inside of that, we have a nucleus right here. So both this soma and the nucleus of a neuron. There's only one neuron on this slide. That means that the rest of these dark staining purple dots, those are our glial cell nuclei. Do also note that we have the beginnings of the dendrites and axons, perhaps, although it's hard to tell the difference, right there, as well as right there and there. And again, based on the stain, we really can't determine if it's an axon or a dendrite. So to identify this, just look for the patterns. Look for a soma, look for a nucleus of this overall larger looking cell that has these dendritic processes or axons, as well as nuclei that greatly outnumber this larger cell. So here we have a sample of nervous tissue. Now, one thing to mention about all these samples we're looking at is they all contain multipolar neurons, meaning they look like this diagram. They have multiple dendrites and a single axon. Multipolar neurons are the most common ones, so that's our focus. However, there are other neurons with different morphology where we only see an axon. So this is an interesting sample. We're looking at 400 power, but it's been cropped, so we're technically looking a bit closer. This sample is central nervous tissue and it's been smeared across the slide to give us this relatively thin and stringy looking sample. So this is a fast blue stain in which the myelin fibers appear light blue and the soma and nuclei appear a bit darker. You can appreciate that some of the fibers are a bit smaller in a different color. So we have myelinated versus non-myelinated, although it's not 100% clear for this sample. Do note that this sample, there are only about 10 neurons, and this was the best sample that we could find out of it. And neurons are three-dimensional, and we smear across the slide, and it gives us this 2D structure. So we lose a lot of the defining factors. So you just kind of have to look at the overall shapes and what we're identifying over here. So that being said, we have our soma roughly right here. And inside of that, right about there is the area of our nucleus. So soma and nucleus of a neuron. Do note we have this blotchy looking appearance. Those are actually Nissl bodies, which are parts of the rough ER that appear darker. And it kind of blends in with the nucleus, makes it a little bit harder to see. Then we also have the beginnings of our neuronal cell processes here. There's at least 3 good ones. So you can see where the fibers continue on beyond those processes. And then we have multiple glial cell nuclei. Those are all of these smaller staining circles. So that's how we go about identifying nervous tissue. Look for the soma, the nucleus, the beginnings of these neuronal processes, as well as the glial cells that outnumber them, and the size difference between them. And that's the 4th tissue type. So along with epithelial, connective, and muscle tissues, it makes up larger organs that ultimately make up larger organ systems and then the living organism. Again, nervous tissues can be difficult to discern, but look for these three parts, as well as the beginnings of the axon and the dendrites, is really key to identifying it.